the factor is ANF factor plays a role. Now we'll be studying after the blood volume is normal, how to stop the release of renin. That is a role of ANF. What is ANF? Atrial natriuretic factor. What is ANF? It's an atrial natriuretic factor. It is antagonist to RAS. RAS will release the renin, whereas ANF will stop the release of renin after the blood volume is reached. Are you all understanding this point?
Ma'am, can you please scroll up?
This we have already studied.
Ma'am, I guess I missed it.
Okay.
Done, ma'am.
Yeah. So now we are studying the role of ANF. ANF is atrial natriuretic factor. It is antagonist to RAS. Matlab, it stops the release of renin. So ANF is a peptide hormone and secreted by the wall of right auricle. Now listen, whenever the volume increase or decrease, but the deoxy blood will reach to the which chamber first? The blood that is moving all over the body, and ultimately where does it reach?
Lungs.
No, heart. Which part of the heart? Which chamber of the heart? The deoxy blood from the body all over the body reaches to which part of the heart? Right auricle. Very good. It reaches to the right auricle by superior inferior vena cava. Do you remember? Right? So it reaches the right auricle. So when it reaches the right auricle, when the blood volume is when the heart will sense the blood that is reaching to the heart is a higher blood volume, the wall of the right auricle releases ANF factor. So when the blood volume increases, there's an increase in blood pressure. This blood goes to the right auricle of the heart, releases the ANF factor of the wall of from the right auricle. Now, what does this ANF do? This will inhibit the juxtaglomerular cells that will stop the secretion of the renin. When there's a stop in the secretion of renin, there's a decrease in aldosterone. When there's a decrease in aldosterone, there's an increase in water and sodium excretion, leading to hypotonic urine and normal blood volume, reaching to normal blood volume. And other side, it will act on vasodilator to get back the BP normal, because in RAS it was vasoconstrictor. Now it is vasodilator which get back the BP to the normal. This is how the antagonistic role of RAS and ANF factor. Did you got the point? How does it work? To understand ANF, you have to be very clear with RAS. Please remember this. Please write down. Which paper should I use? Not test book. Test book. Okay. Next. Okay. Any other paper? I think we have. We did not continue. Darwin and Newton का भी है. Darwin paper is there. Darwin test book. ESIP का कुछ है? Check मैम properly check. Aristotle के लिए offline test है. Aristotle. और Darwin. So it is Aristotle and which one? There are three tests written on Newton also is there. Darwin also is written. So for me it's Darwin, Aris and Phoenix. If you know. हाँ, Aristotle आपके लिए नहीं है. हाँ. So altogether 4 tests है क्या उधर? 3 tests है. Phoenix, Aristotle. हाँ 4 है. Darwin और Newton है. नहीं. हम पेन दे दो. मुझे फिनिक्स का फिनिक्स इसका क्या है? साइलेंट बंद कर दो. छुट्टी किसे दे? Thank God I didn't know how to work yesterday. Huh? I did it for yesterday. But why on 19? Is it done? Done with the writing?
Yes, ma'am.
Any difficulties there? सबसे पहले हम गए थे आयु said them memory and the location all get the pen please ma'am यहाँ पर pen give me the one which writes close by close it's not working yeah and give those results soon okay now did you understood this any doubts now let's move to the function of kidney. The function of kidney involves removal of waste, osmoregulation, synthesis or release of hormone that is erythropoietin. And you know that erythropoietin is a hormone that is released from the kidney and help in the formation of RBCs. That is erythropoiesis. Do you agree with me? Yes or no?
Yes, ma'am.
Please write down. Next, osmoregulation in different habitat. So, if in freshwater environment, what happens? The fish that is in the fresh water, okay, when the in the fresh water, the osmolarity of the fish inside is around 200 to 300 milliosmol per liter, but whereas the water outside that is fresh, which is about 50 milliosmol per liter, okay. So what will happen in that case? More of water enters into the body, more water from outside enters into the body in the case of freshwater species, like the species that are living in the fresh water. So more of water enters. So what are the adaptation? So basically, osmolarity of fresh water is how much? 50 milliosmol per liter, while as the freshwater vertebrates have osmolarity in the range of 200 to 300 milliosmol per liter. I hope you understood. So what are the problems they face? The first problem they faced is loss of body salt to the outside, because salt is inside more, water is outside more, so they lose the salt of the body. Next, entry of excess water, since there is a lots of water, and due to this osmolarity, the more water will move inside the body of the fish. So to live in that condition, there are some adaptation that they come up. First adaptation is to minimize the gain of water and the loss of salt. They have specialized body covered with the scales. Okay, so that the less of water can enter and the more less of ions can exit. So there is a protective covering, that is scale. Next, they pass out a hypotonic urine. Means since the water is entering, a lot of water is entering, they have to excrete that water also out. Say rather than producing a hypotonic urine, they produce a water which is urine that is hypotonic. Next, they also have a presence of ionicides or chloride cells. Means there's special type of cells which are present on the gills, because these cells are losing what salts from the body into the water. So, but these ionicides or this chloride cells will pick up these salts from the surrounding water and give it to the body back. Did you understood this point? Come on, tell me quickly. Yes, please write down.
मार्केटिंग के लिए करो, मार्केट जाके दो दिन फ्रूट लेके आने के बोलो, हैं? मार्केटिंग पे है, ये बटन है, जो छोटे पीस करके बोलो, मुझे आपको नया वाला कोई मिलेगा, Now can you scroll down?
Yes, sure. Write down. Done?
No, ma'am, just a second.
Okay, please write down. Done? Any doubts? Okay. Any doubts? Next. Okay, Shifa, any doubts? Okay, next. Regulation in marine environment. What happened in the marine? In marine, it is exactly opposite. So the osmolarity inside the body is 300, 200 to 300 milliosmol per liter, but outside the salt, the water is salty. It is around 1000 milliosmol per liter. So what happened in that condition is salt will passively move into the body because there are lots of salt outside. So it against the gradient, it moves inside the body and the water comes outside the body. So what are the adaptation? The first adaptation is salts are actively pumped out. The way they actively the ionocytes or the chloride cells used to take up the cells, here they pump out the cells into the salts into the water. Excrete hypertonic urine. Okay, excrete hypertonic urine. Please write down.
Ma'am, excuse me. What is written after that salt?
Passively moves into the body. Salt will passively keep moving into the body. Understood? Done? Done? Next, in the migratory fishes like hilsa and salmon, they keep moving from fresh waters to sea water. So that time, how do they manage? They manage or they maintain the osmolarity with the help of hormones. They manage the osmolarity with the help of hormones. Please write.
Ma'am, can you zoom out a little bit?
Yeah. Done? Done, everyone?
Yes, ma'am.
Any doubts to anybody? Okay. Now, with this, we are ending the chapter excretory system. I'll give you 5 minutes to go through it. Everybody, please go through it. Entire chapter quickly. Just a quick summary. 5 minutes to revise quickly. Done? Is it done? Okay. Anyone, any doubts? Have I shown you the videos on this? Have I shown you the videos on this? Come on, quick. The response should be quick. Yes or no? Don't waste the time. No? Okay. Now do not send me any no. Okay, fine. Everybody listen to this. Okay. Everyone, you have done the revision. I have given you time to revise, Sania. And I hope you all have done the revision. Now listen to the videos.
Human urinary system. The
Just a minute.
human urinary system. The kidneys are a pair of bean-shaped organs, a part of the urinary system. kidneys are dark red, slightly flattened, bean-shaped organs, each about 10 centimeters long, 5 centimeters wide, and 2 to 3 centimeters thick, with an average weight of 120 to 170 grams. The other components of this system are the ureters, urinary bladder, and the urethra. One of the main jobs of the kidney is to filter the waste out of the blood. The urinary system regulates the quantity and composition of fluids by removing metabolic wastes from our body. The system helps in retaining proper amounts of water, salts, and nutrients in the body. The kidneys are placed against the back wall of the abdominal cavity, just below the diaphragm, one on either side of the vertebral column. Their position is slightly asymmetrical. The left kidney is placed a little higher than the right. The outer surface of a kidney is convex, and the inner surface is concave. The inner surface has a deep notch. Here, you can see the ureter, renal artery, and renal vein entering the kidney through this notch. A fibrous capsule surrounds the kidney. Inside the kidney, there are two zones, the cortex and the medulla. The cortex is a reddish-brown layer of tissue below the capsule, whereas medulla is a form of pale conical-shaped striations. The main function of the kidneys is to remove waste products and excess water from the blood. The kidney does it with the help of tiny filters present in it. These filters are called nephrons. The cortex and medulla together comprise millions of such nephrons, all packed together. The nephron is the structural and functional unit of a kidney. Each nephron consists of a renal corpuscle and a renal tubule. A renal corpuscle is composed of two structures: a tangled cluster of blood capillaries called glomerulus, and a thin-walled sac-like structure called the Bowman's capsule, which surrounds the glomerulus. The glomerulus is actually a miniature filtering or sieving device, which consists of a tightly coiled network of capillaries. Waste is filtered from the blood and collected as urine. Healthy kidneys act like a strong filter to make sure the right amount of wastes and fluids are removed from our body. As each kidney makes urine, the urine slides down through a long tube called ureter. Ureters from both the kidneys open into a storage sac known as urinary bladder. Here, urine is stored until the bladder is emptied by urinating. The bladder is connected to another tube-like structure called the urethra for the removal of urine out of the body. Every day, our kidneys process around 200 liters of blood, and around 1 to 2 liters of waste is removed as urine.
Understood? Did you all understood this? You can respond to me now.
Yes, ma'am.
Okay, next. One more.
Excretion. The biological process involved in the removal of harmful metabolic wastes from the body is called excretion. As in other processes, nutrition, respiration, transportation. Different organisms use different strategies to do excretion. In case of unicellular organisms, wastes are removed from body surface into the surrounding water by the process called diffusion. But in case of multicellular organisms, specialized organs are evolved to perform excretion. Excretion in human beings. The chief organ that is involved in excretion is kidneys. The excretory system of human beings consists of 1. A pair of kidneys, right kidney and left kidney. 2. A pair of ureters, right ureter and left ureter. 3. A urinary bladder. 4. A urethra. Kidneys are located in the abdomen on either side of the backbone. Kidney is a basic filtration unit. It consists of very thin-walled blood capillaries. Each capillary cluster is associated with cup-shaped end of a tube that collects filtered urine. Each kidney has large number of filtration units called nephrons packed close together. The waste products are nitrogenous wastes that are collected from the blood in the form of urine. The produced urine passes through ureters into urinary bladder. The urine is stored in the urinary bladder, and the urine is released to outside through urethra. Now, let us learn how urine is produced. The process of urine formation takes place in 3 steps. 1. Filtration. 2. Reabsorption. 3. Secretion. Filtration. The nitrogenous wastes, such as urea, uric acid, are removed from blood in the kidneys. The process of urine formation takes place in nephron. Reabsorption. Some substances like glucose, amino acids, salts, and major water, which are filtered along with nitrogenous waste, are reabsorbed. The amount of water reabsorbed depends on excess water and dissolved waste along with wastes. Secretion. Reabsorbed components of wastes are secreted into the blood when the urine flows through the tubular part of nephron in the kidney. Mechanism of urination. The urine formed in kidney enters into a lung tube called ureter. The one end of the ureter is connected to kidney and the other end is connected to urinary bladder. Urine is stored in the urinary bladder until the pressure of urinary bladder leads to urge to pass urine. When it reaches the pressure to urge to pass it, urine passes out through urethra. How can we hold urine until it reaches to the urge to pass urine? As the urinary bladder is muscular and it is under the control of nervous system, we can usually control to urge to urinate. The process of passing out urine is called as urination.
Clear? Any doubts about the excretory system to anyone there? Each and everybody in the class, is it clear? 38. Only 4 are not there in the class. WSI, just 4 of them are not there. Any doubts? I asked you, do you have any doubts? Because I'm winding up the class. I'm winding up the chapter for chapter. Chapter is done. Now we'll be moving to the next chapter. Okay, Shikha. Others? Yes, that is portion for the 80 marks will be told you very soon. Like, my portion. Mm, start reading structural organization. The chapter will be there. I'll let you know. We all will let you know by this week. Okay? We all will let you know by this week. Is it clear? The test is in Jan and it's on Jan 15th, right? Okay. Next. Next. So, which are the chapters we are done with? Which are the chapters that we are done with? Come on, tell me. Are you all sleeping there? I'm only 3 chapters are left, right? Okay, which are the chapters? Have you done with the biomolecule chapter? Tell me the chapters that we have finished.
Yes, ma'am. We are done biomolecules.
Bila, wait just a minute. Yeah, what are you saying?
Ma'am, biomolecules has been done.
Done. Okay. So, let's move to the next chapter, that is nervous system. Heart is done, right? Respiratory is done. Respiratory system is done.
Yes, ma'am.
Okay. So let's start with neural control and coordination. Neural control and coordination. Just a minute. I'll be stopping the screen sharing and get back to you. 2 minutes. You can revise until then. Are you all there? Okay. In which lesson we have discussed about the nervous system? Tell me. Neural control and coordination. Like it includes neurons. Here we'll be studying the structure of neurons. We have studied already. So here, neurons, how does it work? How do they carry the impulses? And how do we all respond to that? And also we'll be studying the central nervous system and peripheral nervous system. That is brain, medulla oblongata, the different spinal nerves, cranial nerves. All this we'll be studying in this chapter. Clear. To start with, do it now, sometimes later never becomes never. Sorry. Sometimes later becomes never. So let's start this chapter with this quote. Do you agree with this? Do it now, sometimes later becomes never. Right? So whatever you have planned to do it or study today, do it today itself. Do not keep on delaying for some other reason. Yes, my dear students. Next, it is first originated where in the cylinder. Can you all see the screen? Can you all see the screen? The first, it is originated in cylindrata. Simplest nerve system is seen in hydra. Ganglion, that is a group of cyton, first appeared in platyhelminthes. You know what is cyton. Brain-like structure was first seen in insects. Please write down this. Done? Next. So, if you remember in the chapter first, we have discussed about the nervous tissue. Do you remember? Chapter first, we have discussed about the nervous tissue. Do you remember where we studied about the neurons and which are the which are the forms of nervous tissue? What forms the different types of cells present in the nervous system or nervous tissue? Tell me. Which types of cell do you find in nervous system? Neuroglial cells and neurons. Yes, very good. It's neuroglial cells and neurons. These are two different types of cells that are found in nervous tissue. So among this, which is the most in number? which you find in most number, whether it's neurons or neuroglial cells, you find glial cells. Very good. Where? Neuroglial cells. Clear? Now, so we have studied in detail about the which is the structural and functional unit of nervous system in this. Very smart. Which is the structural and functional unit of nervous system? Neurons. Very nice. So, and we have studied the structure of neurons, right? We have studied the structure of neurons. I'm hoping that you know what is cyton, what is axon, what is dendrite, what is axon terminal, right? You know everything. We have studied. You know what is nicel granules, right? You know what is axon hillock. Yes or no? We studied in very much detail, if you remember. Yes? Okay. So today, whenever I give you a test, you should be prepared for that part also. When I say neural control and coordination, you should prepare for, you should include that part here also. Done? Okay. So after thus talking about the neurons and the neuroglial cells, we will move to generation and conduction of impulse. How are these impulse carried in the through the axon, through the neurons? How is this impulse carried? We'll be studying that. So generation and conduction of impulse. Please draw this. Please draw this, everyone. I'll explain you. The outside is ECF, the inside is ICF. ECF is extracellular fluid, ICF is intracellular fluid. Sodium is more outside. Potassium and electronegative proteins are more inside. So inside is negative charge and outside is positive charge. It has sodium channel, potassium channel, and sodium potassium pump. Arrow should be marked as I'm doing. We are talking about the how the impulse are generated, how the message is generated, and it is being transferred across the one neuron to other neuron. I hope you all are getting the point. Yes, I'll explain. I have not yet explained this diagram. Are you done drawing? ECF is extracellular fluid, ICF is intracellular fluid. Okay. Next. Sodium is outside in the ECF, sodium is more. So the membrane is positive, plus sign you can see in the ECF. In ICF, you can see more of potassium and more of electronegative protein. That's why in the inside the membrane, it's a negatively charged. This membrane has two channels, that is sodium channel and the potassium channel. And then there is a pump called sodium potassium pump. You draw this, I'll explain you everything in detail how it works. So please draw and do let me know once you are done. Done? Everybody done? Okay, next. Okay, please write down this. We'll write it down and then I'm going to explain you. Axolemma, you know, it's a membrane of on the axon. That is neurolemma or axolemma is same. It's the membrane of axon. It has sodium potassium pump which is active. Active means it uses ATP. Okay. Outflux means it's throw three sodium, and influx means take in 2 potassium with the use of 1 ATP molecule. It has two channels. One is sodium channel and other is potassium channel, and this both are passive. I'll explain you what is active and passive again. Please write down these 3 points first. This experiment you need to understand, or the conduction of this you need to understand step by step. So write down axolemma has done. So now listen. Axolemma, you know what is axolemma? Look into the diagram. This is axolemma is a lipid bilayer. It's a membrane, plasma membrane of axon. So that is a lipid bilayer. Outside it is called ECF. Inside is ICF. Outside, lots of sodium. Inside, lots of potassium and electronegative proteins. Now, what does it say? We are talking about the pump. It says sodium potassium pump is active. Active matlab use of ATP. Passive matlab there is no use of any kind of energy there. Okay, there is no waste of energy. Now, active means what happened? 3. Outflux means from inside to outside. 3 sodium is thrown away. Can you see the arrow mark here? 3 sodiums are thrown away. And influx means 2 potassium is taken in. And how much ATP is used? 1 ATP is used. And it has 2 channels. One is potassium, one is sodium. And these both are passive. Did you understood this statement? Yes. Is it clear? Others, is it clear? Yes or no?
Ma'am, can you please explain it again?
See, listen. ECF is extracellular fluid, ICF is intracellular fluid. Okay. The axonema means a lipid membrane, which is like a plasma membrane, which is positive outside and inside it is negative. In this membrane or axolemma, there is sodium potassium pump. Look for sodium potassium pump. This sodium potassium pump outflux 3 sodium ions and 2 potassium ions are influx by using 1 ATP molecule, so it is active. Whereas sodium and potassium channels, they are passive. They do not use any kind of energy to move sodium and potassium ions. But whereas pump uses energy to move sodium and potassium ions. Understood?
Yes, understood, ma'am.
Okay, fine. Now, listen to me. This conduction has 3 stages. Okay, it has 3 stages. First is polarized stage, second is depolarized stage, and third is repolarized stage. So everybody please write down this. Write down 3 polarized stage. It has 3 stages: polarized stage, depolarized, and repolarized.
Ma'am, can you scroll down?
Yeah. Very important. Many times questions will be from here. Please write down. I'll explain you each and every point there. Done? Hello. Clear? Done writing? Please draw this graph. Done?
Wait a minute, ma'am.
No problem. Depolarization, then repolarization, then hyperpolarization.
Done, ma'am.
Any doubts to anyone? This will continue. I'll explain you this in the next class. Okay. This is a full mechanism that I need to explain, so I need more time. So other than this part, do you have any doubts? How did you all understood the excretory system? How did you all understood the excretory system? No doubts, right? Okay, no doubts. Prabhu Desai, any doubts? Rishikesh?
No, ma'am.
Um, okay, Shifa, any doubts? Okay, Dia, Sanjana, Saishma, any doubts? Aditi, any doubts? Sanmay, both excretory system and nervous system, what we started. So we'll continue. I'll explain you about this mechanism in the next class. So see you all in the next class. Okay. Take care and bye bye.